The overall goal of the following experiment is to determine DNA regions that are associated with a specific non-coding RNA using qPCR or high-throughput sequencing. This is achieved by cross-linking cells to trap native DNA-RNA interactions in vivo. As a second step, cells are lysed and subjected to sonication to solubilize the cell lysate and shear chromatin into small fragments. Next, biotinylated antisense oligos are added to the cell lysate in order to specifically enrich for the RNA-DNA complexes of interest. Results are obtained that elucidate the identity of non-coding RNA-bound genomic sites based on qPCR or high-throughput sequencing.
The main advantage of this technique over existing methods such as RNA-DNA FISH is that ChIRP allows genome-wide maps of non-coding RNA binding sites at near base pair resolutions.
This method can help us answer key questions in the non-coding RNA field, such as how the ROX RNAs in male flies achieve dosage compensation.
The implications of this technique extend towards therapies of non-coding RNA-related diseases because it allows us to better understand how non-coding RNA regulate chromatin states and gene expression.
For this chromatin isolation by RNA purification, or ChIRP, experiment, 40 million cells will be harvested. After centrifugation, decant the PBS and remove the remaining liquid by carefully aspirating at an angle. Dislodge the cell pellet by tapping the bottom of the falcon tube. Add freshly prepared 1% glutaraldehyde, starting with a small volume, and resuspend the cell pellet. Top up to the full volume and then invert to mix. Cross-link for 10 minutes at room temperature on an